Right.
And somehow the act of sharing my screen kicked me out of Zoom. Oh, the joys. The joys. All right. You guys with me now? Or rather, am I with you? Is really the question. Well, here?
Yep.
Okay. All right. And can you see a chicken egg? Okay. Yeah, my bad. Okay, so movement out of land or out of water onto land involved the development of the amnion. So all amphibian eggs have a yolk sac and a lantois and a chorion, but then you can see that there's an extra embryonic membrane. All of these membranes are called extra embryonic membranes because they're outside the embryo. An extra one here, the amnion, helps to buffer the embryo from environmental fluctuations. And the one of the most challenging things for an egg to get over or survive during the egg stage is desiccating. And so when you have a calcareous shelled egg, like with birds, crocodilians, some turtle species, that's even more impermeable to water loss than a parchment shelled egg. The soft shelled eggs that are common in snakes and lizards and most of the turtle species. But having a shelled egg with an albumin layer, a chorion, and an amnion basically provides enough membranes between the embryo and the outside world that it really slows down the rate of desiccation. If you ever work in a zoo or are propagating amphibians and reptiles at home, one of the things that you will find yourself challenged by when you have an animal that reproduces and you get eggs off of it is incubating those eggs not only at the proper temperature, but also at the proper level of moisture that gives them enough moisture that they don't desiccate, but not too much moisture that things like mold and stuff like that begin growing on the surface of the eggs. And so, so moisture when you move out of water onto land is a crucial aspect of that transition. Is just avoiding desiccation. We've talked about also how the structure of the skin changes as you move from aquatic environments to terrestrial environments. The epidermis becomes thicker, multiple layers. They become keratinized, more impermeable to water, etc. And so there are a variety of adaptations that occurred all in concert with one another that allowed this transition, but certainly the structure of the egg was one of them. And so oftentimes you'll see books referred to the amniota, which are all of the vertebrates that contain an amnion, and that includes all of the reptiles, the birds, the mammals. So these extra embryonic membranes, the amnion, protection and cushioning of the embryo. Also, it's one more aqueous layer between the embryo and the outside world. The yolk sac contains the energy that has been provisioned by the female to the offspring. The allantois is where the nitrogenous waste are stored. It's kind of like an extra embryonic urinary bladder, if you want to think of it that way. And then the chorion is used in gas exchange, and then there's some sort of eggshell. Remember that the amphibian eggs are not shelled. They stop with essentially the egg membrane on the outside, but there's no hardened or parchment shell to their eggs. You can see right into them, which is one of the things that makes them nice for developmental biology. You can actually view the development of the embryo through the egg. Other things about the amniota. In the amphibians, in the vast majority of amphibian species, fertilization occurs outside the body. So the female will basically extrude the eggs, and then the male will spray sperm across the eggs, and so you have external fertilization. Because the eggs in reptiles are shelled, you have to have fertilization occurring of the egg before you wrap that egg up in an impermeable shell. And so one of the other things that happens with this transition to terrestrial living and terrestrial reproduction is that you have to get the sperm to the egg before the egg gets wrapped up in a shell. And so with this comes the development of copulatory organs, usually exhibited by the male, that allow the male to achieve internal fertilization as opposed to external fertilization. And we have a couple of different ways in which this occurs. The development of penises with erectile tissue, which occurs in mammals, turtles, crocodiles, and some birds. Most birds don't have an erectile penis. They have a cloaca, and they do this behavior that is referred to in the bird literature as a cloacal kiss, which is basically them putting both cloacas up next to one another and transferring sperm that way. The tuatara, the Rhynchocephalian, this weird lizard-like species that occurs in New Zealand, has lost its penis. And in the case of snakes, the penis has become modified into what is known as a hemipenis. And so this is just the phylogeny of the vertebrates from your book that deals mainly with the reptiles, figure 2.7. And what I've shown is the gain of an erectile penis mapped onto the phylogeny, the secondary loss of the penis, which occurs in birds and also occurs in the Rhynchocephalia, and then the development of hemipenes in the squamata. So these are how these characters would map onto this structure. So it looks like the hemipenis of snakes and lizards did not develop from the erectile penis of all of the other mammal and reptile groups. It was actually lost in the common ancestor to all of the what's known as the Lepidosauria, the snakes, lizards, and the tuatara, and then was developed independently, this hemipenis, which when we see a picture of it, you'll be like, that looks nothing like what I think of an erectile penis looking like. And you'll see that that arose independently in the lizards and the snakes. So this is kind of the evolution of erectile organs that allowed for internal fertilization in these amniotes, these land-dwelling organisms. So you can see on the phylogeny that the taxonomy is on there. So the Lepidosauria is this common group of both the tuatara and the lizards and snakes. We have the Archosauria, which includes the birds, a bunch of extinct dinosaurs, and the extant crocodiles and alligators. We have the Testudines, which is the turtles, and then we have the Synapsida, which includes all the extant mammals and the extinct non-mammalian synapsids. And we'll talk about what synapsid and diapsid means here in a little while. It has to do with structure of the skull. Well, we'll talk about it on the next slide, actually, as it turns out. So on the skull, there is an opening where the eyes rest, the optical foramen or orbit. And then on the back portion of the head, there are openings typically. And you can lack those openings altogether, which is what the anapsid condition is, and this is what occurs in turtles. You can have the synapsid condition, which is what occurs in mammals, and then you can have the diapsid condition, which occurs in all of the other reptiles and birds. And the diapsid condition can have two full openings in the skull, or in the case of lizards, there's a single opening, but there's a second opening that has actually just been broadened to the point that it's no longer closed at the bottom. So it's still a diapsid organism. It is just it's just that the second opening has gotten so large that it's actually reduced the total bony components of the head. These bones, these openings are essentially where muscles go, and in many cases, these muscles are the chewing muscles, and so chewing muscles that connect the lower jaw, which is not pictured here, to the skull through these openings to allow a broader area of attachment, and it provides a more straight up and down movement of the jaws as opposed to the movement that pulls it around the curved surfaces of the skull. And so that's a pretty major modification of the brain. So for a very long time, people have used the structure of the skull as one of the characteristics in determining phylogenetic relationships among different taxa. And so this is what it looks like in kind of a diagrammatic form. Here are some anapsid skulls of early amphibious organisms. This is a diapsid skull, and here is a synapsid skull from Dimetrodon. So these are fossil representatives of anapsid, diapsid, and synapsid. And then these are some modern representations. Here's an American alligator. You have these two fenestrae here. This is where the eyes are, and this is what makes this a diapsid individual. This is the skull of the tuatara, and you can see once again it's got the upper and lower temporal fenestrae. This is the skull of a turtle, and you can see that it does not have those kinds of openings. This is the skull of a fossilized reptile, and once again you have the upper and lower temporal fenestrae here. So diapsid, 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 anapsid. So that's what those terms mean. And when you go back to the phylogeny, you see that the synapsids are all of the mammals, one temporal fenestra. The diapsids are all of the extant reptiles except the turtles, which are on their own branch over here, because they presumably came from a diapsid ancestor and then lost those two openings. But it includes everybody in the reptiles except the turtles, which are anapsid. So birds, crocodiles, snakes, lizards, tuatara. So there are two big groupings, the Lepidosauria and the Archosauria. Lepidosauria are the snakes and lizards in the tuatara. The Archosauria are the dinosaurs and birds. Birds presumably evolved from lineages of dinosaurs that have long gone extinct. One way of thinking about dinosaurs is that some of them went extinct because the Earth was hit by a meteor that killed off most of life, and the ones that didn't go extinct in that event essentially evolved into birds. And then we have what's known as the Crurotarsi, which are the crocodilians. And this actually is now in their own group of organisms called the Crocodilia. Crurotarsi is a upper level taxonomic grouping for both ancient and extant crocodilians, but all the extant crocodilians are in Crocodilia, as you can see in the phylogeny. So you will hear me using some terms that I've now used for a little while without telling you what they mean. What did I do with my bag of tricks? And I'm not sure if I'm saying these clearly enough. We can refer to extinct organisms as organisms that lived on the face of the earth at one point in time and don't live here any longer. And then we can have extant. And these are organisms that are currently living. And so I'll refer to extinct and extant, and hopefully you can hear the differences between those terms when I use them. So there are plenty of crocodilian representatives, but they are extinct. And then there's this other branch of the Crurotarsians that is also long extinct. But then there are a limited number of crocodilians that are extant, that are still with us. One of the other things that you will notice in this phylogeny, so I've now gone back a couple of slides, is that there is this boundary between the Cretaceous and the Tertiary. And one of the things that you see at this, what's commonly known as the CT boundary, is that you see all of the kind of dinosaurian lineages and early Lepidosauromorpha lineages go extinct right there at that hard juncture. And this was presumably when this meteor crashed into Earth, killed off the vast majority of terrestrial organisms at that time, and only a limited number of lineages survived that event. So you see this hard break: Plesiosauria, Ichthyosauria, Ornithischia, Pterosauria, other saurischians. They all die out at that boundary. And the way geologists or paleontologists kind of set these boundaries is these are oftentimes set long before we knew a lot about the history of the Earth. They're basically determined in many cases by the kinds of fossils that you find in strata of certain ages. And Kara knows about this, right? You're working on fossil plants?
Yep.
Yeah. So, you know, you'll see some fossil plants that occur in some strata and they don't occur in other strata, and presumably that's because they were alive at one point in time and then they went extinct between one set of strata and the next set of strata. And as it turns out, there's a layer of soil, a layer of rock in the sedimentary rock that corresponds to this boundary. And as it turns out, it's made up of a bunch of elements that are not commonly found on Earth, that are commonly found in meteors, which is why the people have put forth this hypothesis that it was actually a meteor strike that led to the Cretaceous Tertiary extinction event. Later on this semester, we'll talk about extinction events. There have been a number of major extinction events over the course of time, but that's later in the semester, because we're currently in an extinction event today, which is caused by human activities rather than meteors hitting the Earth. So the major groupings. The Lepidosauria includes the lizards and snakes, as well as the Tuataria. The Tuatara, which is in the order Rhynchocephalia. They have a cloacal slit that goes across the body, as opposed to a longitudinal cloaca. They've lost their penis, but they have replaced it with a single penis with hemipenes, which are basically a paired penis. And these guys have a regular cycle of shedding. And so they will generally shed either large sections of their body, scales, or entire sections that come off all in one sheet, rather than having what we do, which is we're constantly shedding the outer layers of our skin on a daily basis. Have you guys ever seen the movie Gattaca? No? Oh my gosh, this is a movie that all biologists, I thought, watched. It's about this dystopian future where your fate is determined by whatever your genes say it should be. So they give you a genetic test as an embryo, and then they can genetically engineer you so that you will get a better job and have better social outcomes, et cetera, as you are as a fetus. And then that sets you up for whatever your fate is going to be. And one guy who was genetically inferior decides that he's going to buck the system. So he pays a genetically superior guy to give him his blood for blood testing and things like this. And every day before he goes to work, he has to scrape like all of the loose skin off of his entire body so that he's not shedding cells at work, because they go through at work and they vacuum up your keyboard and then they test to see if that's actually really you or not. It's a twisted dystopian future, but we're constantly shedding skin cells, and that's not something that lizards and snakes and Rhynchocephalians do. Their skin comes off in big sheets when it's time to replenish the outer layers of their epidermis. The case with crocodilians and turtles is that they tend to shed their skins in little chunks over time. So bigger chunks than us, but smaller chunks than lizards and snakes. So I have a Madagascar giant day gecko at my house that I collected in Florida a number of years ago. And when he sheds, his whole body comes off in one big piece for the most part. And you can usually tell that he's about to shed because he gets duller in coloration. And then on the day that he sheds, he goes pretty much almost all gray because his outer scales have become separated from the under scales. And then when that stuff starts coming off, he basically starts reaching around onto his back and stuff and twists himself around. And he actually consumes his skin when he goes to shed. But he goes from kind of dull brown to gray to vibrant green, like over the course of an hour as he gets rid of his entire skin. The Archosauria include two groups, the Crurotarsi, which includes the crocodilians, and the Avametatarsalia, which are the pterosaurs, dinosaurs, and birds. These guys have stronger cranial musculature than, for example, the lizards and snakes do. The lizards and snakes often have what is described as a kinetic skull. It is a skull that is more loosely articulated. It's more flexible. And that's largely so that they can eat prey items whole and basically wrap their head around them to a certain degree. Snakes have a much more kinetic skull than lizards do, but then lizards have a more kinetic skull than, for example, birds do. They're also stronger in their cranial musculature, which is probably correlated, and they have different teeth. The crocodilians, obviously birds don't have teeth at all, but the crocodilians have thecodont teeth that are firmly set in sockets. And their pelvic girdle is very different from the pelvic girdle structure of the other reptile groups. And it's the pelvic girdle structure that was kind of the first sign to scientists thinking hard about the evolution of vertebrates when they began to think, okay, maybe crocodiles are more closely related to birds than we thought that they were. So this is a typical vertebrate arrangement of the pelvic girdle, where you have the ilium, the ischium, and the pubis. They can become fused and form arcs as they are in humans, but they basically exist as kind of independent pieces. But in birds and crocodiles, what you see is that there's this extension of the pubis that becomes fused and goes off in the same direction as the ischium. And so this pubic ischial fusion here is a characteristic that we see both in crocodiles and birds, and it's not seen in the other vertebrate groups. And this actually changes the kind of the way the pelvis is situated, and it also changes how the legs get attached to the pelvic girdle. And it's very different than what we see in the other vertebrate groups. So this is a huge synapomorphy that unites crocs and birds in this Archosaurian lineage. So presumably all of these archosaurs have this pelvic structure. And you also see this pelvic structure in dinosaur groups as well. And so that's probably one of the single most important things that groups these groups of organisms together. The Archosauria also have what are known as pneumatic bones. This is why you don't feed chicken bones to your dogs, actually. And the reason is that as a weight-saving adaptation, the bones, rather than being solid like most of our bone mass is, they have this this relatively thin outer cortex, and then you have these trabeculae, which are kind of like structural support struts that span across the bone, which allows them to maintain structural strength while also reducing weight, because most of the mass of the bone is actually just empty space. And so when you think about the girders on a building, or when you see these big cranes that are construction cranes, you'll see that their structure is made up of cross struts between, relatively thin tubes of metal. And that allows those structures to maintain their strength while also reducing weight. And so this is what a pneumatic bone looks like when you look at it in a longitudinal section. And once again, you'll see that this is the way the bones of crocodiles are structured, and it is also the way the bones of birds are structured. And in birds, the lungs actually have outpocketings. So the birds have taken this idea of pneumatic bones to an extreme, in that you have a bird. This is the most terrible looking bird in the world, but it has respiratory passages that go down to a lung, but then there are these outpocketings from the lung, these air sacs that come out of the lung. And what this allows birds to do, because they need a lot of oxygen because they have really high metabolisms in most cases, is when they breathe in, the air goes into the lung, and then it goes through the lung into these air sacs. And these air sacs don't have any respiratory surfaces. They're not used for respiration. But what they do is they send the air all the way through the lung, and then they push the air all the way out through the lung, so that you don't have any dead space at the bottom of the lung like we have in humans. When you breathe in and breathe out, there's always some air in your lungs that you can't expel because you just can't push it all out. This is what makes you float when you're swimming, or when you've had your body dumped after being murdered, which is why you have to weight a body down. Not that I've ever weighted a body down, but I'm just saying. I'm joking, by the way. So one of the things that this gives birds is it gives them ability that when they're pushing air through their lung, it's always relatively fresh air, and there isn't this dead air that just kind of sits around and loses its oxygen content over time. And these air sacs, you see when you look at the structure of a flighted bird, you'll see that these air sacs actually go through the bones of the body of the bird. And so this trabecular structure of the bone actually allows for that routing of these air sacs through the bones. And for birds that are flighted, it really saves them a ton of weight. They can still have good, strong structural bones at a fraction of the weight. And this is, of course, why bird bones shatter when you break them. And then they become sharp little splinters. And if you feed bird bones to your dog, they get stuck in their throat. And that's not good, but it's all because of this characteristic, which is once again a very strong synapomorphy in support of the Archosaurida. You'll never look at chicken bones again, probably, in the same light. Turtles. Now, turtles are a problem. And turtles are a problem taxonomically because they have undergone extreme morphological change and modification. And so here is a tortoise that has been cut sagittally down the midline so you can see inside of it. And what you can see is that these are the vertebrae up here at the top, and they're all fused together. In the dorsal part of the vertebra form the dorsal part of the shell, and then these lateral plates that form the bulk of the dorsal portion of the shell, these are actually just flattened rib bones that have all grown out, flattened, and fused. You see a real reduction of the skeletal structure inside that is supporting the limbs, and much of that skeletal structure has once again also become fused to both the carapace on top and the plastron below. The plastron is then again made up of an expansion of part of the ribs and the sternum. And so turtles are complicated in figuring out where they fit in with other groups because they've undergone this pretty extreme body modification over evolutionary time. They're the only group of terrestrial vertebrates that lack temporal fenestrae, so that becomes kind of an odd apomorphy for the turtles, so it's not really useful in placing them in a group or relative to other groups because it only occurs in that one group. And then, like I said before, their body form makes it difficult to assign homology and figure out, okay, well, what aspect of the body of this organism might be a synapomorphy that groups it with other groups of turtles, with other groups of reptiles, rather. And so as a result of this is that depending on whether you're looking at morphological data or whether you're looking at molecular data and how those data kind of fall out, the turtles are always placed in different places in the phylogeny of the vertebrates. In some cases, they don't really know where it is, and so it forms what we call a polytomy or a polytomy, where this is an unresolved branch of the evolutionary tree. And then in other phylogenetic reconstructions, they appear as the sister group to the crocodiles and birds. And that is actually the phylogeny that I believe your book takes, where you have the Testudines as the sister taxon to the Archosauria. But then other authors have looked at data and found that the Testudines, which is the turtles, should be the sister taxon to the Lepidosauria, the snakes, lizards, and the Tuatara. And so one of the tricks about doing an evolutionarily based taxonomy is that in order to do an evolutionarily based taxonomy, you have to know what the evolutionary history of a group of organisms are. And unfortunately, the turtles, because of their structure, are systematically not well placed, and as a result, their upper level taxonomy is not as well worked out as these other groups are. Because they're just so different. So that's a quick romp through taxonomy and systematics. What questions do you have? Let's get rid of the terrible bird picture. Okay. So the next thing that I want to do in class over the next couple of class periods is give you a brief introduction to the different groups of organisms that we're going to be looking at. And so what this is going to be is essentially kind of a tour, a global tour, if you will, through the major groups of amphibians. And I just want to preface this by saying we're not going to cover all of the groups of amphibians for a couple of reasons. One, there are just a lot of them. Two, if you don't become a world traveler, you're never going to come into contact with many of these. And so it's going to be a tour of amphibians or reptiles that mainly concentrates on New World groupings and also concentrates on North American groups of organisms. But I will occasionally take some asides to other parts of the world with organisms that are either interesting biologically or they have some interesting sort of convergent similarities with organisms that live here in North America. So we'll start with an overview of amphibians, kind of what makes an amphibian, and then we'll kind of go through and introduce you to the various families. Hopefully some of these families you're already becoming familiar with, because we are looking at, for example, the mole salamanders, the Ambystomatidae in lab, and the Plethodontidae in lab. And so hopefully this will dovetail nicely with what we're doing in lab. There are also going to be situations where I draw your attention to salamanders that are in other parts of the United States that are of interest for one reason or another. But I'm not going to spend a lot of time talking to you about old world representations of these different groups. So we'll spend the rest of today and probably sometime on Monday talking about amphibians. Then we'll do a similar thing with reptiles. So that's what's coming next. Questions? All right. Can you guys see the PowerPoint? All right. Just checking. So when we think about amphibians, we've already talked about the fact that they have complex life histories. They start out as an egg. The egg is aquatic. Typically, they typically have aquatic larvae, and they typically have terrestrial adult stages. One of the things that makes sometimes studying herpetology difficult is that this complex life history that I've given you, once again, it occurs in most but not all amphibians. So not all amphibians have an aquatic larval stage. Some amphibians have a terrestrial larval stage. Some amphibians go through their entire larval development inside the egg and hatch out as little baby froglets. And so you're going to see this most but not all at the end of a lot of these generalities that we make. And it's important to recognize that not all amphibians do the same thing. And it's also important to know what some of the exceptions to the general rules are. And so we'll talk about that more when we talk about reproduction and life histories later in the semester, but we'll also cover some of that today. Amphibians generally are thought of as having a permeable skin, although the permeability of the skin varies a lot within amphibians. Some amphibians have really, really permeable skin and other amphibians don't. So in the specimens that you're looking at in lab, what are some groups that seem to you to have more permeable skin and some groups that have less permeable skin? That was a question. What are some groups with less permeable skin?
It seemed like all the salamanders, like as a group, had less permeable skin than like some of the frogs I was looking at yesterday.
Did you say less or more permeable?
More permeable.
Yeah, more permeable. Yeah. So, yep. So many of the salamanders are much more closely tied to water than the frogs and toads are, and so they will occur in cool, moist areas. Many of them will spend large portions of their lives in water. Give me some less permeable. What's like the least permeable group of animals you're dealing with in lab?
Would the wood houseye one be one?
Yeah, so all those bufonids, all the members of the genus Anaxyrus, all of the toads, are even more impermeable than, for example, the tree frogs and the chorus frogs, which have thinner, more permeable skin. And so they do vary somewhat, but they're much more prone to evaporative water loss than, for example, reptiles, where the skin is modified into a set of relatively impermeable scales. So two things about this using skin as a respiratory organ is it's nice because it allows you to breathe through your skin, but the downside is that you tend to suffer evaporative water loss at a higher rate. And because it's permeable, you are not only capable of losing water across that permeable membrane, but you're also capable of absorbing anything that is in the environment through your skin. And so you tend to be more sensitive to things like environmental contaminants and things like that than reptiles do, for example, or mammals for that matter. So for this reason, frogs and salamanders are having difficulties now. Part of the problem with having this nice, moist respiratory organ that is the skin is it tends to allow you to absorb pollutants. And so if there are pollutants in water that can kill you, you will absorb those. In some cases, people have found that certain pesticides basically lead to, basically they become estrogen disruptors. They essentially mimic estrogen compounds and basically turn male frogs either into female frogs, or they interfere with the sexual development of male frogs to the point that they're not really reproductively capable. Another thing that happens because of this permeable skin, because it's moist, because it's thin, it also becomes an avenue for things like fungal infections to take root in the skin of these organisms. And so we have a variety of funguses that are generally known as chytrid funguses. And these are fungi that basically infect the skin of the amphibians. And the chytrid fungus is responsible for a number of recent extinctions of organisms, especially in the tropics, but even here in the United States. There are some species of frogs and salamanders that seem to be relatively tolerant of these chytrid funguses, but a lot of them are not. I went to collect salamanders in Oregon a number of years ago, newts in Oregon a number of years ago, and my scientific collecting permit was denied because I applied for it in like February. And in January, there was a new rule that went into effect that prohibits the trafficking of any amphibian, regardless of protected status. You're not allowed to carry these amphibians across state lines. And the whole reason for that is to reduce the spread of chytrid fungus by picking up infected individuals in one place and possibly releasing them in another place. I actually had to get an injurious wildlife permit from the U.S. Fish and Wildlife Service. And part of that process was that I had to escape-proof my lab. I had to demonstrate to them that I had made modifications to the lab such that if an animal, so I had to do three things. I had to make modifications to how I stored my animals such that the likelihood of one of them getting out of their individual cages was reduced. And then after that, I had to demonstrate that the room that they were in had modifications to the doors and windows and things like that, such that if they got loose in the room, they couldn't exit the room. And so the room where I keep amphibians or reptiles, the shelves are relatively tightly spaced so that you slide the containers in there with the amphibians or reptiles in them, and they're very close to the lid so that the lid can't just be popped off by the animal. And then they're stored inside of an individual room that has metal strips along the floor so that if somebody does get out, they can't flop onto the floor and then crawl out under the door. And then there's a larger room outside that then leads out to the hallway, and that door also has metal strips at the bottom of it. So when I went to apply for this injurious wildlife permit, I had to essentially escape-proof the lab because of problems having to do with these skin funguses that are killing off amphibian species. And since then, there are also reports of fungal diseases that have started plaguing snake populations in particular. And so oftentimes you'll see professional herpetologists out in the field collecting animals, and they'll be wearing rubber gloves or nitrile gloves. And after they've handled one animal, they'll take those gloves off, they'll put on a new pair of gloves before they handle the next animal, once again, just to keep disease transmission down. So aquatic reproduction, oftentimes these individuals lay their eggs in water, usually but not always. These eggs then hatch out into an aquatic larval stage, usually but not always, and they will develop in the water until they go through metamorphosis, where they transform from the larval stage, the larval form, into an adult stage, and then they live out their adulthood as terrestrial adults, usually but not always. So there are exceptions to every one of these things. So for example, in the tropics here in the New World rather, there are a group of frogs that build foam nests. And what happens is the male and the female get together, they copulate. As the eggs are coming out of the female, the male is on the back of the female, and he's kicking his legs in such a way that it kind of develops the mixture of sperm and eggs into a froth. That froth becomes adhered to whatever plant they happen to be sitting on, and the outer layers of that foam nest harden up in the air. And when they harden up, they become impermeable to water loss, and then the frogs are developing in the eggs in this foam nest. As the frogs develop, they eventually hatch out, and then as they hatch out, they release the contents of their eggs. That then begins to moisten the foam nest, and then the foam nest starts to develop holes in it, and then the tadpoles dribble out of the foam nest and fall into the water. So their eggs are not laid in water, they're actually laid in a foam nest that is usually outside of the water, but then the aquatic larvae drop into the water, where they then develop as normal tadpoles. Poison arrow frogs, frogs in the genus in the family Dendrobatidae, they will, at least in some cases, lay their eggs in water. The tadpoles will hatch into water, but then the parent, and in many cases this is the male parent, will then go and pick up the tadpoles. He'll basically put his butt in phytotelmata. Phytotelmata are water-holding areas of plants. So if you see a bromeliad, bromeliads are plants that usually are found growing on another plant, like a tree, and they have leaves that kind of come out like this. A pineapple is a type of bromeliad. And inside these whorls, these whorls basically form a depression that holds water. And what poison dart frogs will do is they lay eggs, and these are called phytotelmata. And because these phytotelmata hold water, there's a whole community of organisms that lives in them. And what will happen is the dendrobated frogs lay their eggs in there. After the eggs hatch, the tadpoles will consume whatever food is in the phytotelmata. But once they're finished with that phytotelmata, if they don't get out of that phytotelmata and move to another one, they'll basically just starve to death. And so the parent will come, dip their back end into the water in the phytotelmata. The tadpoles will squirm up onto the body. I don't have a picture of it. They'll squirm up onto the back of the frog, and then the frog will go hopping through the forest, find another bromeliad, stick his butt in the water in the phytotelmata. The tadpoles will slither off. They'll occupy that phytotelmata for a period of time. Then they'll pick them up again and move them to another place. And so part of the parental care in dendrobated frogs is actually the tadpoles living part of their life outside of water on the back of the parent. Where metamorphosis is different is that, so for example, in many Eleutherodactylus frogs, they have what's called direct development. I think that's maybe on the next page. Yeah, direct development, where the eggs are laid on land, usually under the bark of trees, in leaf litter and things like this. And the tadpole goes through all of its development inside the egg until it reaches the froglet stage, has 4 legs, it's lost its tail, et cetera, and only after it reaches that stage does it actually hatch out of the egg. In Puerto Rico, I have been looking for frogs before and pulled the bark off of a tree, and when I pulled the bark off of the tree, I uncovered a mass of eggs of Eleutherodactylus coqui, the coqui frog there in Puerto Rico, that had gone far enough in their development that they were just on the cusp of hatching, and the disturbance of pulling the bark off the tree caused enough disturbance on the nest that they all hatch at once. And so you pull off the bark, and then a bunch of little froglets jump out into your face because the act of disturbing them caused them all to hatch en masse all at once. And so direct development is when you're going to do all of your development inside the egg and then hatch out as a little miniature of the adult form. In the case of caecilians, there are some direct developing species that retain the eggs in the oviduct. They give birth to live young, and in the case of the caecilians, they actually live for a time period in the oviduct, and they actually feed off of the lining of the oviduct before they are finally born. And then we've already talked about paedomorphosis, this idea that because of heterochrony, you have different tissues of the body developing at different speeds, resulting in reproductively capable individuals retaining larval characteristics. And so this is what we saw back in the last presentation with the axolotl. We also have species here in the United States that do this as well. There are a number of species of plethodonid salamander down in Texas associated with the Edwards Aquifer that are paedomorphic. They have external gills, they're fully aquatic, but they're also fully reproductively capable. So the skin is super important in these guys. It is a stratified squamous epithelium, but it's never keratinized. It doesn't ever, the outer layer doesn't ever lose its nucleus and become compressed the way our skin does. And as a result of that, it keeps this, this permeability, which once again, our skin does not have. And then there are a bunch of glands in the dermal layers that produce mucus and produce granular glands. And this mucus actually coats the skin, helps the skin ward off things like bacteria and fungal infections. And then the granular glands are actually the glands that ultimately produce toxins. And so there are going to be two types of glands in the deeper skin, the dermal layers. Remember that your skin, or you may not remember this, maybe you never had a course where they covered this, but you basically have a thin epidermis at the surface, and then that is underlain by the dermis. And then you have things like these granular glands that produce toxins, and these mucus glands that produce the mucus secretions that coat the skin. And you can see that if you go all the way out onto the very surface of the skin, you can see that all those epithelial cells have nuclei in them. And so this skin is much closer to the skin that is on the inside of your lip. If you take well, you don't take histology because we don't teach that here anymore. But if you were to take histology, you would have looked at a cross section of the lip, and you'll see that it's the thinnest layer of stratified squamous epithelium on the lip, but there's just a very thin layer of keratinized epithelium on the lip, just a very thin layer, like 1 or 2 layers thick. And then as soon as you cross over from the outside of the lip to the inside of the lip, it transitions from the outer layers being keratinized, where they're full of keratin and the nucleus has been extruded, and you go to the inside of the lip where it's moist, and all of those top layers have nuclei in them. So your inside of your lip is much closer in structure to the skin of an amphibian than the rest of your skin on your body. So this is kind of what well, this is exactly what it looks like. This is amphibian skin. So you can see the epidermis, and then you can see the deep dermis. And then at the bottom of the dermis, you can start to see muscle layers, which are now into mesoderm. In developmental anatomy, did you guys go through the different germ layers and things like that? Or developmental biology? One of you took that, didn't you? Who took developmental biology? Was it Kelsey?
Yeah, I think it was Kelsey.
Oh, it must have been Kelsey, yeah. So continuing with the skin, some species go through the process of estivation. This is kind of like a hibernation, but rather than being a response to cold, it's a response to a drying environment. This is a spadefoot toad. This unfortunately is not necessarily one of the species that is really prone to dry environments. This is a Couch's spadefoot that is in the southeastern United States, but all of these spadefoot toads go through estivation. And what they do is they bury themselves underground during dry periods. And when they get down there, they will then secrete mucus around their body. The outer layers of that mucus dry and harden and create this impermeable barrier that then allows the mucus that is around the body to stay moist. And they'll stay down there, in some cases, for years and years and years, until there's a big torrential downpour, torrential rainfall event. Water trickles down through the soil. When it gets down to the level where the toad is living, it will then begin to soften that outer mucus coating that is dried. It starts to change the chemistry of the mucus that is around the frog. The frog senses that. He'll break through his cocoon, burrow up to the surface, hop off to a pond where there are tens of thousands of his conspecifics breeding. They will breed. They're called explosive breeders because they breed in these gigantic mating aggregations. They'll breed, they'll leave their eggs in this pond, and then they'll leave the pond, hop back to where they came from, burrow back into the ground, and then sit around and wait for the next big rain. The eggs are then left in these temporary water pools, and these temporary water pools tend to dry very quickly, and so they tend to have larvae that develop very quickly. And in some cases, they'll actually morph into different morphological forms, where one of the easiest ways to get out of a quickly drying pond is to just eat other members of your population, so they become cannibalistic. They develop bigger jaws, they develop these kind of teeth-like structures in their mouth, and they'll actually consume other tadpoles in an effort to grow fast enough that they reach metamorphosis size before the pond dries up. And here's a video of a spadefoot toad burrowing. Let's see if this works. I never have any luck with this. Can you guys see YouTube?
Yep.
Okay. So this is the plain spadefoot. This is a spadefoot that we don't have in the collection, but lives here in Missouri. And he's in a terrarium here, but he's moving his back legs back. This is a spadefoot toad. He has a cornified spade on his back legs. He's just pushing the soil to the side. And as he pushes the soil to the side, he just slowly descends into the soil. So you can see his feet working, pushing the soil out to the sides. And in this case, he will only burrow down until he hits glass on the bottom of the tank. But they can be feet into the ground when they do this out in nature. We had, when I was in grad school, we had a herder's spadefoot in the lab, and we would always have to just pick up the aquarium and look at the bottom to see if he was still doing okay, because he was always buried down in the soil on the bottom of the tank. So we'll talk about the reproduction in spadefoot toads in more detail once again when we get to that portion of the course. Go away. But explosive breeding, fast development times of larvae, and then large periods of the adult life is actually just spent hanging out underground waiting for it to rain really hard. This is also one of the reasons why you don't find spadefoot toads in collections very often, is you have to kind of be at the right place at the right time in order for you to encounter them. And we're at 10:05, so I guess I'll just stop there and we'll pick up here on Monday. Questions? Quinn, you still coming up to look at specimens today?
Yes, I am.
Okay, I'll run upstairs right now and open up the lab for you.
Okay, thank you.
Sure.